come to in layman's terms, and with Guy Nohra, we try to take issues out there that are usually a little wonkish, and we make them simple, concise, to the point, with a little bit of common sense. Our guest today, actually, I know, and I have spoken to him before, so I'm really looking forward. We come from the same world. I've had several different, as the audience knows, we've been talking about all these things that have been happening in around the world, and there's always a crisis somewhere. So we're doing this, but now finally we can have this conversation. So Jacob, first of all, pleasure to have you. Second, it's great to talk to you again, but now in this format, and I can't wait to have our audience hear what you're doing, why you're doing it, why it's going to make a difference, what it matters. When we're talking, if you use acronyms or any words like this, I will probably stop you and have you define them for us. Because even though I might know them, that's not the case with our audience. But first, you're a serial entrepreneur. I've always been impressed about how in your previous company you did not take any investor money from people like me, which I always find I should feel bad, but I don't. I actually feel great for you. So I think that's another thing. And yeah, please tell us about yourself, about you grew up, you decided to take this track in life, and then through your first company. And I really want to know what's happening with your current company, because I think that the subject of a universal vaccine is fascinating, especially after the last couple of years. And we might or might not go there. We'll see how this discussion goes.
All right. Well, first off, it's great to see you again, and thanks for having me on. I'm really glad you're doing this podcast. So just for your viewers, hi, everyone. I'm Jake Glanville. I'm the founder and CEO of Centivax, previously the founder and CEO of Distributed Bio. Prior to that, I worked at Pfizer. My background is entrepreneurship and computational immune engineering, which is using computers, statistics, and mathematics to analyze and engineer immune responses. And that's taken a number of forms, from the antibody discovery work to make drugs at Distributed Bio and similar work I was doing at Pfizer, and now with universal vaccines and the universal antivenom.
So I'm someone who just for the first time in my life heard computational immunology. So let's for the layman, what does that mean, in a way that they can go, oh yeah, that makes sense. We can talk about it at the kitchen table tonight.
So from the dawn of man, our bodies have had a problem, and that is that it takes us, even in ancient times, 15 years or so to reproduce. And these days, 20 to 30 years to reproduce. But meanwhile, the pathogens that attack us, they can reproduce in like 30 minutes. And they can mutate and evolve every time they do it, which means our pathogens evolve way faster than we do. And the way that we have survived against this ever-changing foe is that our immune systems have devolved this ultra complex method of producing huge combinatorial, like mathematical diversity of antibodies and T cell receptors and NK cells and cytokines. And by constantly shuffling a deck with a finite genome, we're able to confront these rapidly evolving pathogens. Now that's really cool, but it also requires math and computers to understand it. And so what computational immunology is using big advances in the genome's arms race, these laser-based rapid cell sorting technologies and other technologies that let us look at millions of data points coming out of our immune system, and then to be able to interpret it, to decode it using algorithms and mathematics, so that we can understand some like pretty obvious things that you think we would know, but it's only recently that it became available. Like after you get a shot, how many antibodies do you produce? Or why is it that sometimes our immune system messes up and it starts attacking ourselves and creating autoimmunity? Why does a cancer evade our immune system, and how can we overcome that? How can we make a universal vaccine? And so these are practical things that we want to build, but sitting between those two is this combinatoric system, and mathematics is how we're going to decode it.
Okay, so here we are. We have our body. we're going to use this a little bit of a war analogy. The pathogens are the soldiers from the other side. They attack us, but they're much more flexible than we are in general. So our body now, over all these years, has developed a way of dealing with this, which is we have special forces in different
Yeah.
areas that go back and fight whatever change a pathogen has made, or if there's a new pathogen that comes
Yeah.
along.
Yeah.
Okay,
And
good.
normally that works great. The problem is that the pathogens are often changing all the time. So our immune system remembers. So once we win a
Yeah,
war, all the soldiers tell each other, hey, when this comes back, remember, if you see those guys, start shooting. The problem
yeah,
is a lot of these pathogens are like a rogue army that's constantly changing their outfits. And so you kind of remember, hey, if you see a bunch of guys in like red hoodies, remember those guys were attacking us last time, but then they show up next time wearing kimonos. And so the immune system has to learn all over again. That's why we need new flu shots every year. That's why the
yeah.
coronavirus vaccines like stopped working and they had to make new ones. And
Okay.
so the work that I do is try to train the immune system to recognize the parts of the pathogens that they cannot change. So you basically, after a while, see if you're like, okay, they change their outfits, but they're always bringing guns. So they can't change the
Okay.
gun, otherwise they're not dangerous. So here, stop looking at the outfits. Just if you see a bunch of guys showing up, no matter what they're wearing, if they're carrying a bunch of guns, those guys are a problem. And if you can teach your immune system to do that, then you're protected no matter how they change.
Okay. So you're looking for the constant in
Yeah,
the attacker, whatever that might be, and then now your immune system is not going to be fooled by a red
ex-
hoodie or a kimono. It's going to say, I'm following the one thing, which is a gun, which I know is the way that they're going to attack me.
that's right.
That's
And
that's
yeah,
perfect.
and
Okay.
then and it's the thing that they can't change because they're no longer useful if they don't have it, right? And if a bunch of guys
Okay.
show up with no guns, you don't care. And the viruses are like that. These pathogens are like that. They have tricks, but they have a finite number of tricks that have worked so far because we didn't have this golden age of biotechnology. So this is kind of a remarkable period in history where we have these tools that would allow us to win a forever war against some of our most ancient enemies. And we're making pretty exciting progress. So we have our universal flu program. We just raised an oversubscribed $45 million Series A led by Steve Jurvetson at Future Ventures. This guy
Sure.
sits on the board of SpaceX. He's just a phenomenal ability to
Sure.
go recognize early huge wins. I think his investments have, I think, resulted in $1.4 trillion in
Okay.
value generation, which is more than the national annual GDP of the majority of nations on the planet. And so
Yeah.
I'm really excited to be working with him. And then in
Okay.
addition to that, our same technology, we recently published a Cell paper that got international attention on using the same biology to go after a universal antivenom, which is a single cocktail to work on the 650 species of snakes. And so there's really a
Okay.
real significant change in medical science that we'll be able to affect with these universal immunity technologies. And we're working at the forefront of it.
And tell me quickly about, of course, no trade secrets or anything, but
Sure.
tell me about the technology and the science that you are using, which has now finally made it to where we are, that's allowing us to fight that next battle much better equipped than we ever have been, which is your company, which was
Yeah.
oversubscribed. Congratulations. And I
Thank you.
remember Steve from the Draper Jefferson's days and even before that. So I'm that old, but yes.
Thanks.
Uh,
Sure. Yeah, so I'll tell you what we did different, and I'll keep that same analogy going. So for the last 200 years, since Edward Jenner invented the vaccine technology, that really hasn't changed that much since its inception. What we do is we create a vaccine for a single strain, and so we expose the body to it. And the body, I told you all these cool things it does, but it's also pretty dumb in a certain way, and that is, if you expose uh, a vaccine with a single strain, it just randomly makes antibodies against the surface. It doesn't know what's the most important. So, example, if
mhm.
you're trying to train your troops to watch out, in essence, what the vaccine's doing is you're hauling out a bunch of guys in red hoodies and be like, all right, remember these guys really carefully because they might be a problem. And the problem is your troops don't know what to pay attention to, and so some guy's going to look, oh, okay, I'm going to look for a red hoodie, I'm going to look for a red jacket, I'm going to look for a little like a logo, and so they fixate on a couple random things, and they don't know that the gun's important. They don't know the gun is what's going to remain in common, and so
Yeah.
then that immunity wears off because what they learned about is no longer useful when the kimonos show up or the big silly hats. And so that is why we have to keep training the soldiers over and over again by creating two new flu shots every year. And we've been doing that since the 1940s. And still, by the time you make it, the virus has already changed. So a lot of the antibodies you've made are useless, and they have to update them every year. And even then, it's only 10 to 60% effective. So this last season, 1 in 3 people who got a flu shot benefited. And that's a huge problem, not just medically. It's like a Pavlovian problem, because you have a lot of people out there who are like, I got the flu shot and I still got sick. And it's really hard to communicate that to people. And you have to go, like, it's a losing battle if you're having to go into the weeds to be like, statistically, on average, you're going to have a reduced rate of severe disease. Like, no, that's not how people experience life. They want a shot that makes them not sick.
Yeah.
And so the way our technology works, because people tried a number of things. They tried trimming parts of the protein, or they tried mutating things. Their goal was to try to reduce the amount of distractions. So they're like, let's say if we let's bring guys in that have like short sleeve cutoff hoodies or something. So they stopped paying attention to the arms, and it just wasn't enough to help people focus.
Yeah.
And so I did the opposite. I was like, no, I want to make sure they just focus on what matters. And so what we do is we took a century of influenza all the way back to 1918, the pandemic that killed more people than all of World War I and World War II combined, and one of the 5 pandemics in the last 100 years. And we take just a tiny amount of 22 different viruses. There's not enough of any one of the viruses to produce an immune response. So if you vaccinated any one of the ones in my vaccine, nothing happens. But they all share those common sites. So it's kind of like us being like, all right, soldiers, we're tired of this crap here. We're going to roll out 30 dudes. And they're all wearing totally different outfits, but they all have guns. And we're going to put them far enough away from you that the little details get lost. And so when you look at all 30 dudes together, it becomes immediately obvious, like, oh, wait, gun is the thing they have in common. And so then they remember the proper thing. And so that's the fundamental basis of our technology. The reason no one did this before was that it was like fundamentally really counterintuitive to deliberately put too little of something in a vaccine for it to be useful on its own. The assumption for 200 years was, all right, well, the first thing you got to do is make sure whatever we put in, there's enough of it that it's individually going to be targeted. And I violated that on purpose. That's what our IP focuses on, is we put too little of each strain so that you can't produce a response to a single strain. But they share a couple common sites. And so the B cells that are pumping through your veins and mine right now, and everybody has them, but they're rare. The ones that recognize the shared site, they see 20 times the dose as all the B cells that recognize the strain specific. And so like most of your B cells, they're antibody producing cells, are like, I don't know, I don't think I've been vaccinated. I'm not really seeing a signal around here. Meanwhile, those B cells that are going to make those awesome broadly neutralizing universal antibodies, they're like, I don't know what you're talking about. There's all sorts of protein, because they get 20 times the dose. And so you're only vaccinating those broad B cells that recognize the key site. And that was the fundamental breakthrough.
I see. So this is very important. And again, we I would love to get into the weeds, but I won't, because that's not what people want to spend time on. But everybody knows that gets the flu vaccine every year that it's kind of a guess. They kind of go, oh, you know what, we think this year is going to look this way, and they have a committee that decides what they think the most likely thing is going to be. And then I don't know the number. You probably know better than me, but it protects you maybe 25% of the times, or some percentage like this.
It's like 10 to 60% is the best it ever is. This last year it was 35%, and it's probably going to be worse this coming season. So this is something everybody knows. They kind of make a guess every year, but you kind of assume they're making a guess for things that are maybe, because it takes them 6 months to make it. So you're like, oh, maybe it's 6 months outdated. No. It is multiple years outdated, because when that community meets, they need to balance not just what are the strains that are likely to come, where they're just guessing, but they also need to balance the manufacturers being able to produce the stuff. And there's a risk that if you pick strains that are too recent, like it's 2025 right now, you'd think they'd put 2025 strains into the vaccine. No. Yeah. The current vaccine that people are going to take this fall has a 2021 strain, a 2022 strain, and a 2023 strain. These are all old, highly outdated strains, but they leave them in because they're like, we think they have enough sites in common that they should still work. And we're trying to balance the manufacturers reliably being able to produce those things, which have been well characterized and can be produced, against trying to update it just enough that it's not totally useless. And so, of course, if you imagine, that process is going to result in consistently outdated vaccines. When they say it's a match,
Sure.
it never means identical. It just hopefully means not that bad that it's not useless. And sometimes that low bar is not even satisfied.
Yeah, it's less useless than so why well, so the average person listening is going to say, well, why don't we use the 2025 2024 strains in order to develop this year's vaccine?
Yeah. So the reason they
What's
don't
the
do
end?
that, yeah, back in 2004, they had a huge problem where they tried to update some newer strains, and then some of them you couldn't produce it, you couldn't manufacture it. And so that what they ended up with is a horrible shortfall of not enough vaccine for global need. And so they operate in a more conservative fashion now, where they check and they go, well, we think this vaccine still has some power. It's still working. We know it's deteriorated, but it's a risk if we try to update to a 2025 strain. What if that strain, the pharmaceutical companies, we tell them put this in, and then they're like, we can't make it. And so for that reason, they usually give multiple years of time for the pharmaceutical companies to go test and make sure that they can grow large batches of the material that goes into the vaccines. And that's it's this regulatory cycle and making sure you can actually make the stuff, which is a really weird. It's unusual, right? Most of medicine, you spend time making it once, and then you can just mass produce it,
Sure.
as needed.
Yeah.
This is a product that only exists for 6 months. You basically make it for a season for the Northern Hemisphere. You rush to produce it at risk. You then try to sell what you can. You try to, and the four companies make these weird calculations of how much to make based on how much their competitors are making. And you don't want to make too much because the product is useless by the end of that season.
Yeah.
And then you do that whole thing all over again 6 months later in the Southern Hemisphere. That's the current state. And it's the best we've had so far. Look, I take flu shots. I'm an asthmatic.
Yeah.
But I'm taking it as like a statistical insurance policy. What I'd really like is to take something and be like, cool, I'm not going to get sick for the next, 2, maybe 3 years. Eventually I'll get a booster, but I know I won't get sick. I don't have to work. I'm a CEO. I don't have time to get sick. And then the more profound consequence of a universal vaccine, which once it's commercialized, it'll just be called the shot, the flu, right? The flu shot. Is that you no longer have pandemics of flu. We've had 5 in the last century, and everyone's worried about this H5N1 thing. They're like, is it not going to keep infecting humans? Like, you don't worry about any of that anymore. Once this is out, it also protects against pandemic ingress viruses from other species. And so there's no longer oh God, what do we do? Like, there might be an H5N1 outbreak. There's just like a reminder from the CDC, hey, if you haven't had your if you haven't had your Centiflu booster shot in the last couple of years, why don't you go in and take one, because there's some H5N1 that's infecting people. Like, the pandemic era is over with this technology.
Okay, so I want to definitely talk about the technology, but I'll give you a quick anecdote. I used to not take the flu vaccine because I'm a science numbers guy, and I'm like, well, so I'm going to take a vaccine. So maybe 22% of the time I don't get sick, and maybe I'll just take a bunch of zinc and quercetin and see what happens, right? And then I'll never forget 2000. I got very sick from the flu. I mean, very sick. I was like 2.5 weeks, and then postpartum clinical weirdness and all that kind of stuff. And I was like, okay, I'm getting the vaccine every year, and I became a believer. Because 22% protection is better than 0, given what happened.
That's why I take it.
So your technology. Why? I remember when I first heard of what you were doing, I was like, that sounds too good to be true. Because if you tell me you're going to have something that's going to work for everything, and really have 100% protection. When it comes to the flu, it's like everybody should have that, because it makes so much sense. Because the flu, of course, with immunocompromised people or older people, they can die, and the young ones like us, hey, we spend 2 to 3 weeks feeling awful. So the technology, again, without divulging, of course, anything that's your own trade secrets. What have you discovered to actually have that potential?
Yeah. So first off, I'll say the history of medical science is that this conversation has happened over and over again until the change took place. So people said the exact same conversation we're having now, they had in the early days of research on antibiotics. Everyone was like, this is too good to be true. It will, because humans have a hard time imagining a world that's different than the one that they're living in. And so the whole,
Mhm.
if you look back, it's fascinating reading where everyone's like, there's no way. And they had a bunch of theoretical reasons why it would never work to create a broad spectrum antibiotic. And then they came out and they changed medicine. And in a similar fashion, our antivenom program, I spent 7 years on that, where everyone's like, there's no way, it's not possible to create broad spectrum antivenom because each snake is different, just like each strain is different, and so it'll never happen.
Sure.
And then we proved it. We published a Cell paper, and those same people are coming back, and now they're having a different conversation. They're like, okay, well, how do we manufacture it? These other issues. And so
Okay.
I think the punchline here is that medicine improves, and if you look at the history of medicine, we can make these kinds of breakthroughs. Now, that said, I want to create some parameters around it. So we don't think 100% is where we're going to land. So I look at the history of all vaccines, like the best vaccines. You're talking about 90-95% protection. Like there's this remaining stubborn 5% of people who get a shot where they might be very immunocompromised or have a genetic deficiency, or for whatever reason that it's hard to get to 100%. So I think we're looking at somewhere in the target product profile is like 80-95%. And that's still a huge game changer. But I just want to say, I think there is this small minority of people that take any vaccine where their titer response is just not optimal. But that's a huge jump from something which is a roll of the dice where you lose most of the time, which is what currently we have. And then the other thing I want to constrain it is, we call it a universal vaccine, but it's universal as to flu. So it'll provide extremely broad protection against all flu, but we need to apply the same technology, but it would be a different vaccine to go after like HIV or malaria. And it's just because they don't have any site in common.
Okay.
The flu
Okay.
is the guys with the guns. The HIV is a bunch of bears in the woods, right? They're just totally
Fine.
different things. They don't share a
Yeah.
common thing.
Fine.
So what we've done, my kind of the success of my previous company and then this one, is that I basically relentlessly hammer on just building a really strong foundation of bioengineering and overwhelming data. And my rationale is like, humans don't live that long in the grand arc of history. And so I don't have time to go repeat things. I want to measure twice, cut once, and just really overwhelm. Like when my army goes across a river, I build 3 bridges. And so we do very careful, disciplined engineering. And when it comes to vaccines, I'm always paranoid that if you pay too much attention to one animal model, like you're like, oh, it looks perfect in mice, then you get shocked when you go into humans, and humans are not a mouse, right? And like that's been a problem in medical science. We've cured cancers in mice all the time that don't translate into humans, for instance.
Sure.
And so the way I defended myself from that is we used mice and rats, but we also used pigs, we used ferrets, we used cows, and we even used something called human immune organoids. And we tested this across all of these systems, and all of them are showing universal immunity. So there's some common thing that is not unique to a given immune system. And even we tried different versions of the vaccine, and they all had the same operating phenomenon of universality. And the reason you like pigs is that they actually get the flu, and they're the source of most pandemics. So what happens is a pig can get infected with
Yep.
bird flu and pig flu and
Amen.
human flu, and the RNA shuffles up, and so that makes a horrible new virus, and that's what pandemics come from frequently. They're also a $180 million a year flu shot market. So I felt that there could be a separate vertical and a veterinary application, as well as like, look, if my goal is to go get rid of pandemics, I damn well better treat humans and also pigs, because then you can push the problem further out to the birds.
Sure.
So the reason you choose ferrets is that historically ferrets have been, they say, oh, well, ferret lung tissue is very similar to humans. Okay, kind of. The real reason is that everybody's been doing ferret studies in flu. And so by testing in ferrets, you also can compare to, 80 years of research.
Okay.
Cows, because there's this H5N1 outbreak. And I was like, let's go immunize some cows. We're going to see H5N1 protection. And so we're preparing a publication showing that. And then the human immune organoids was this company called Parallel Bio, where, I do all these animals, and I don't want to rest. I want to de-risk everything I can before you pay the big bucks of manufacturing, which we're in right now. And then the phase 1. Because I think a lot of companies, they're focused too much on their timelines and milestones, and they skimp on the early stuff. And I think that's why you'll get attrition when you get into phase 1 and phase 2, and then they're shocked that because they've been spending too much time on hype and on hitting milestones, because I think business people with no technical background sometimes are leading these things, and they don't realize the risks. Where my view is that biotechnology is kind of like poker. Like, yeah, being able to read people and be charming, that stuff matters, but so do the numbers. And you need to know both in order to win the game. And so so I tried all these animals to basically make myself as confident as possible, but still I wasn't satisfied. And I heard that there was these advances in human immune organoids. This is where they take an organ donor, they take out their lymph nodes, which are a key. That's that thing that swells under your armpits and your neck and
Sure.
so forth.
Yeah.
It's the party house where all of your immune cells come and they coordinate to make antibodies and coordinate some T cell coordination, and some other cells come in there. So these guys take those from organ donors, they shuffle them up, and they basically make a bunch of these little mini lymph nodes in a petri dish. And you can make a bunch from a single person, and we can vaccinate them. So this is an adult with adult immune memory that we can vaccinate. And in fact, we can vaccinate the same person now with different versions of the vaccine or different doses or competitor vaccines. And we can see how that immunity plays out in a human before you start your human trial. And that's a pretty profound and useful thing, because there's certain parts of the immune system that are very human specific. And so we ran these studies with Parallel Bio. That the early technology was developed at the Mark Davis lab at Stanford while I was a grad student there. I have to admit, I'm a little embarrassed because like it was happening and I was like, oh, what are they're screwing around with tonsils? Like, all right, it's kind of cool. I missed it. And then, I like to think of myself as a forward, but I didn't fully value what Mark was building, and Lisa, who was and then later I was like, oh, this is gold. And so I connected up with Parallel that had commercialized the technology, and we ran these awesome studies. We were able to go deliver our vaccine and find this universality induced in human memory compared to the other vaccines that didn't do that, and learned a whole bunch of other useful things. We're using that technology repeatedly now.
Okay. So I can't tell you how hard it is for me to not get into venture mode and stick to layman mode, but I'm trying. So there's so many questions I want to ask, but I won't. So to the to our people out there, if we look at your process, um, you're in preclinicals right now.
Yeah.
You have the basic thesis and scientific thesis in place. And when will you start your human clinical trials?
Yeah. So we're in the CMC, the GMP manufacturing process, which is a very expensive, detailed, and necessary process where we rather than the lab where we can give vaccine to animals and so forth, we check to make sure it's sterile, but we move relatively quickly. There's QC. There's a whole different level of QC and quality assurance when you produce a drug for humans, because you need to make sure you're going to make that when you run a study, what you're learning about in that study is the exact same product that you're going to end up giving to people. It's incredibly documented, and you're going to make it the same way every time. So that seems like a good idea. You should do that. It's also super expensive, and it's subject to wedding/funeral pricing, because everybody knows people are in a rush. So the negotiation is extra obnoxious. But to be fair to these guys, they have to run these things in like clean rooms with people wearing special suits to go make your drug. So we're in the middle of that right now. In about 8 months, we're going to start our phase 1 study. And so this is a big deal for us. Normally, a phase 1 clinical study is you're just getting mostly safety and maybe you'll learn. But in our case, we also get efficacy out of our phase 1. And that's one of the reasons we chose flu as the first indication for our platform, is that because we've been making new flu shots since the 1940s, and they needed to make 2 new flu shots a year, they've come up with a way to very quickly assess whether a flu shot is working well enough against a strain. And that's this gold standard correlative protection. It's a blood test where they can take blood from a person and then they test that against some virus in a petri dish, and it's called HAI. And basically, if you can take your blood and dilute it 40-fold and it's still able to block the virus from doing something in the petri dish, it tells you that you likely are protected. And that's pretty phenomenal, because that means in our phase 1, we'll get safety. And this thing is highly likely to be safe. If flu is the most studied vaccine, because we've been replacing it since the 40s. And we're using a chemistry which has been de-risked in billions of people. And so we think that the safety is, and we've run the preclinical talks but the more interesting thing is we can take the blood from all the people in the phase 1 studies and we can test them. And we're not just testing them against 3 strains, like which is the normal flu shots. We have a panel of 24 strains, including 2025 strains, the ones that are actually going to get people sick, 2024 related, very similar strains, We're going
Okay.
to test the guidance. We're going to test old pandemics. We're going to test H5N1, the current thing that might be a pandemic, and we're going to test
Yeah.
a couple years that were horrible mismatch years. And so we want to establish universality of efficacy using a correlative protection in the phase 1, which is basically proving that this vaccine would provide that universal protection. And you get it in phase 1 because of this assay. So this is a big deal for us, and we're looking forward to it.
Right. And will that be the first time that someone is this broad on their phase 1?
Radically so. So there've been efforts to create universal vaccines. This is a big
yeah,
request. I mean, like Trump
sure.
signed an executive order in 2019
Yeah.
calling for a
Yeah.
universal flu vaccine.
Okay.
And then recently, McCary and some other folks at the FDA are basically saying we want to see one in the next 5 years. Everybody
Okay.
wants a universal flu vaccine. So far, the technologies that were deployed were basically like, okay, here's the antigen. Let's just try chopping parts off, or let's try blocking parts. That thing I said didn't work. And so people went in and they ran, and most of the techniques they came up with were not eligible for the HAI assay because getting into the weeds a little bit, the
Yeah.
spike protein on flu, it's called hemagglutinin. It kind of has like a head and a stem. And the antibodies that are active in the HAI assay are hitting the head, and the head mutates even more than the stem does. And so most groups
Okay.
just gave up. They're like, it's too hard. And so they
Okay.
tried other techniques where they couldn't use this accelerated approval process. They couldn't use the HAI. And so they ran phase 1s where it was more traditional. They got safety. They got a little glimmer of like, oh, maybe we're seeing some antibodies that look good. And then they got disappointed in phase 2. Or they didn't even try antibodies. They were like, I don't know, it's just too hard. Let's just try T cells. And people have tried that. And so this is going to be by far pending success, provided that the adult humans do the same thing as the adult immune organoids, the pigs, the ferrets, the cows, the mice, the rats.
Yeah.
This is going to be a stunning breakthrough, and we'll be able to see clearly whether or not we achieve that in this phase 1.
Okay. So for people out there, if you're still with us, and I hope you are. So phase 1 is going to be safety. Phase 2 is going to give you a little bit of efficacy, and then you run a big phase 3 clinical trials to really confirm efficacy. And after that, you got hopefully FDA approval, and then it's available to everybody. So Jacob, how many years are we looking at before I can tell my friends that they're going to have your vaccine that's going to be universal, and don't worry about anything anymore? You're not, you're 100% close to having immunity on the flu.
Yeah, so it's accelerated, right? So we get that efficacy in phase 1.
Yes.
And then the other advantage is that we get, there's this thing called accelerated approval for flu because they had to go make new vaccines come out quickly. And there's a history of other new premium vaccines. These are vaccines that work a little better than the standard flu shot. They're for older people because their immune systems don't respond as well. And they've used this path. So this path is pretty fast. You don't have to have a 30,000 person trial. You just have to have 3,000 people for safety. And so we can compress and accelerate. So that said, we're still looking at about 2029 is roughly when this would be available on the schedule that we currently have. And that gets us through.
Hey, that'd be perfect because I'll still be in the range where it can help me. I know I'm sounding selfish here. So that's awesome. I want to, you and I reconnected very briefly recently on AI. So people want to know, what's AI going to do to biotech? So in 5 minutes, can you tell me what AI is going to do with biotech?
Sure. Yeah. So here's my complicated thoughts on this. On one hand, I sound like a grouchy old dude because there's a lot of hype out there, and I'm an engineer. I'm like, no, a bunch of the stuff people are saying is not true. On the other hand, I think there are ways it could be useful, but I think it's kind of like the .com bubble. It wasn't, and I had a conversation recently with someone about this, and I'm like, that makes sense. It's like all that stuff that happened in the .com bubble that burst, it ultimately came true. It just took another 10 years to mature all that stuff, right? And so I think we're in like the AI hype bubble. I think AI, it was so good at these large language models, even though they have problems. And then AlphaFold was objectively a stunning success. But the problem is AlphaFold didn't just use a large language model. It used a huge amount of structural data of general structures, and it solved a general problem. And so it satisfied the requirement of a huge amount of data to gain inference on patterns. But that gave rise to this false assessment that like everything in biotechnology could be solved right now through AI, which is just not true. And so like I looked at for instance, I just did my Series A, and my wife asked, Like how many series errors are happening right now? And so I searched for it. Almost every company is an AI based company right now. I think we're in the middle of a bubble. I'll tell you what, like where the technology is right now. There were companies claiming, "Oh, we're going to solve antibody discovery with AI. We're just going to have the AI tell us what antibody." Now, that would be cool to have a
Yeah.
computer. You can press a button and it makes whatever antibody you want. It's also not real. So there are a couple of companies doing this, and an article came out that I was invited to give review on, where there are a couple of companies claiming they had an AI engine to do de novo generative design. And in fact, if you looked at their patents, what they actually did was take other people's published antibodies and they made a couple small modifications to them. And so
Yeah.
they had to backtrack and say, oh, well, that's the first step. But it really, to me, it was a little bit of a bait and switch, or maybe a lot of a bait and switch. And the reason is, unlike structures, of which there are huge amounts of structures in PDB and even more representative homologs, related sequences, which is AlphaFold used, antibodies, you don't have any of that. You have like one antibody that binds here is a totally different universe than an antibody that binds here on the same protein. It's because it's a surface binding another surface.
Yeah.
And so uh, it was just a much harder problem. And I think that people are going to have to work a lot harder there than they were anticipating for a success. Now, there are some successes. People are coming in and
Sure.
creating,
Sure.
binders of like 50 nanomolar or so with, if you search enough. Now,
Yeah.
a lot of that work, they actually had to use a library in between. So they use a model to come up with like thousands of designs, and then they put them on yeast
Yeah.
display and go, oh, we got some hits. And I was like, well, did you run the control experiment where you just randomly created 1,000 antibodies, put them on
Yeah.
library, and
Yeah.
then check to see if you got hits? Like, tell me that, did you do better than random chance? I think
Yeah.
they are a couple real deal. There's a couple companies out there, a couple,
Yeah.
Baker's the real deal. I think
Sure.
they are exceeding random noise, but I still
Yeah.
think that the quality of the molecules, I think it's going to be real hard to do affinity maturation entirely in silico. I just anticipate that. But that said, here's where I think what they're building now could be useful. If you can write a piece of software that can spit out, I don't care, spit out 96 antibodies, if 10% of them hit the target, but you can get those basically as fast as you can synthesize and express them, even
Mhm.
if they're not very potent. Let's say they're 15 nanomolar molecules.
Yeah.
You just have a really powerful ability now to go test biology quickly. So you can create a surrogate molecule with extremely quickly to test hypotheses against various epitopes and various targets. Then that justifies doing the hard work, which I still think you have to do to go make a
Sure.
high quality molecule. I also think they have to be antibodies. So, I did this universal antivenom program, and another paper came out, which was really cool, but they created these random protein folds, these generative folds. They're helical bundles, and they're like, hey, look, these
Yeah.
things bind
Yeah.
these sites. Our antibodies were much better because they came from a human who'd injected himself for 18 years with 16 different species of venom from around the world. And so they were affinity matured to be like these incredible picomolar binders.
Yeah.
But the other problem is that one of the big problems with snake antivenom is that it's horse antibodies or sheep antibodies. And so when you inject those in a person, you get a whole bunch of side effects because your body
Yeah.
detects that it's
Yeah.
foreign. Well, these AI random generated molecules are going to have all those same problems most likely because they are totally non-human. They look like alien protein, and so they're going to be very
Yeah.
immunogenic. And so I
Yeah.
still feel that, like for biologics transformation, you need to be able to have an AI engine generate a high affinity molecule or appropriate affinity molecule while the developability properties of a self-looking protein is preserved. And so I think that people are working on that. I don't think it's
Yeah.
intractable. I just think, caveat emptor. I think there's a lot of groups out there
Yeah.
that are saying they're doing this that aren't. And I think there's a limited number of groups that have more work ahead of them than they were anticipating. That's my feel.
And I really appreciate this because again, this is exactly the kind of answer that my audience needs to hear. Folks, it's exciting. It's gonna happen, but it's not happening tomorrow
Yeah.
when it comes to drug development, and it's okay because these things move slowly. So again, it's really important because this except I mean you see it everywhere I go. I say, AI is going to change the world, going to change the world. But in this particular sense, it's going to help the world at this stage, but we're not changing it right now, and it's going to be a while before we do. And I think it's again, it's very important for people to know that. I don't want to downplay your venom. Did you want to talk about your venom program? Because I know that's also something you're working on, and I didn't mean to downplay it at all.
Oh,
I think it's pretty cool actually, because I live up here. I'm up here in Reno, and there are snakes everywhere, and I'd like to have your venom in my back
yeah.
in my kitchen, just in case.
No, the conversation was just right. So, like, look, our lead program is the universal flu shot. It's a $7 billion market. It's a huge global problem. It creates pandemics, and it sets the stage for us to demonstrate the technology, which will then just bam, bam, bam apply to other pathogens. So, for sure, that was the right thing for us to focus on. It's what was the
Okay.
basis of this $45 million raise. The
Okay.
Antivenom project, it's kind of the other end of the spectrum. It's the same biology. It's how do you create universal immunity? But it's the exception is I found an edge case of how I told you most people's immune systems don't recognize the conserved sites. I am always fascinated with what are the exceptions? What are those extreme phenotype individuals that somehow broke that rule and succeeded? Because I think you can learn so much about vaccine science, antibody protection, if you can find such subjects. And so we have this program that has been supported by the NIH and in collaboration with Peter Kwong at University of Columbia prior to the NIH, where I was interested in universal immunity in 2017, and around the same time, the World Health Organization was like sounding the alarm that companies were starting to discontinue antivenom products, and the reason they were doing it is that a lot of people, it's 140,000 people die every year, 300 to 400,000 lose a limb, but most of them are poor people in the developing world, right? Who don't have a lot of infrastructure and frankly, they don't have a
Sure.
lot of money. And so,
Yeah.
and the other problem was not only is it a smaller market, it's maybe 500 or $600 million a year. So it's not $7 billion a year. That
Yeah.
market is currently fractured into 40 different products that each
Yeah.
treat a single snake or a couple snakes. And so most of the 650 snakes don't have an antivenom for them. And for the ones that do, it's basically a public service that these companies are largely producing these materials.
Okay.
And so I got interested in it, and I was like, well, I don't know. Nature's pretty lazy with what I've learned from these viruses. I wonder if antivenom, if snakes probably, even though they're all different, they probably have like a limited set of toxins that they all use, even if they're mutated. And maybe they have these shared sites, like I'm working on for flu and HIV and so forth.
The famous guy with the gun
Yeah,
versus the
exactly.
hoodie.
Yeah. I'm
Yeah.
like, I
Yeah.
bet they all
Okay.
have a gun, right? Because they all have to do a toxic task. I bet they can't
Yeah.
change that surface. And so I looked, and sure, and we live in this amazing age where I can just download structures and alignments of a bunch of toxins that are in databases. And I checked, and sure enough, there was like the gun, there was the conserved patches. And so I was like, I bet we could find a broadly neutralizing antibody against a couple of these toxins and create a universal antivenom that would just like in one fell swoop, it would unfracture the market. So then you'd have at least a $500, $600 million market, and it would have much broader, greater protection. If you notice, it's the same thesis as my vaccine technology. And so I wanted the antibodies to be human, because the other problem with antivenom is that it's horse antibodies or sheep antibodies. In fact, they chop them in half. It's Fab2, because that makes them a little less toxic and create less anaphylaxis and serum sickness. But the side effect is that now they have a short half-life. So you have to keep administering this stuff every couple hours to the patient. If you even have an antivenom for the snake that bit them, and if that antivenom is inappropriate geographically, because the snakes are genetically diverse, even in a species. And so I was looking around for like a clumsy snake researcher, and this is like a very side idea. And then suddenly I find this guy who, his name's Tim Freedy, and for 18 years, he had decided to establish universal immunity to snake venom. And so he had injected himself with escalating doses, starting with micrograms and building up to milligram doses of cobras, taipans, kraits, Western diamondback rattlers, mambas, like 16 of the most gnarly species around the planet. He built up immunity, 654 documented immunizations with meticulous records, and then ultimately 202 bites over a series of years from snakes that absolutely should have killed him 202 times.
Okay.
And his side effects were some swelling and pain and mostly not much else. And so I heard about this guy, and everything I had mentally modeled about the immune system, like the hairs on the back of my neck went up, where I was like, if anybody's established this universal immunity to snake bite and has the secrets of a universal antivenom pumping through their veins, it's Tim Freedy.
That's the guy.
And so I called him up. I had to get in touch. He basically had some videos on YouTube, but otherwise he had no internet presence. I was like, this guy's a purist. And so I contacted these reporters that had done some stories on him. One of them put me in touch, and I called him up, and I'm like, hi, I'm Jake. There's no easy way to say this. I would love to get my hands on some of your blood. And he was expecting, he's like, I've been waiting for this call for a long time. I wanted to get studied. And so we started this collaboration to search through, to use phages, these technologies I had in my laboratory to rapidly search through his immunity to try to find these universal, broadly neutralizing antibodies. And we started finding them. And we started working with Peter Kwong. Same story. Everyone was like, that won't work. Epitopes, people were like, epitopes don't matter. Broadly neutralizing antibodies aren't a thing. And then the last few years, that's all been changing. We published a paper in Cell maybe a month and a half ago, where we demonstrated we have a cocktail that covers half of about 300 species of all venomous snakes, and we're working on the second cocktail. So the universal antivenom is within reach, and I think it just goes to show the power of universal immunity and how it, from antivenom to influenza, this is going to be a new class of medicines, which is going to profoundly change human health experience and for our generation, our children's generation, and every generation that comes after. Like antivenom barely changed in 125 years. Vaccines haven't really changed that much around the strain-specific thing in 200 years. But this is the generation where we can change that, and I'm excited to be part of that.
That's awesome. So I'm gonna end with a slightly controversial question here because we're talking about vaccines. I know a sizable piece of my audience has been, I guess, for lack of a better word, disappointed in the latest management of the COVID vaccine situation, for lack of a better word. And and if you follow the numbers, there's more vaccine skepticism now. I'm not a don't do the measles vaccine on your kids, but I'm also, I want to see the side effects, and I want to net positive when I'm looking at the next vaccine, given everything that's been going on in this world. I personally, of course, when you come up with your universal, probably I look at your research, make sure that it's safe, and it's safe for my family and my mom and my kids and so on, and I'll adopt it because I know you, I trust you, but I also can make my own mind. But there are people out there that unfortunately have been disappointed by the way we've handled this situation, and they're going to be very skeptical. Now I need them, if what you're doing has the promise of what I think it does, I need them to actually, I need to bring them back. And I need them to trust us. How are you thinking about that whole thing?
Yeah. So first off, I want to acknowledge some of the concerns that they have. So I'm the son of hippies. I grew up in Guatemala around a lot, like hippie expats and Mayans and stuff. So I think I came a little bit of an outsider of some of the common perspectives in our industry. And I felt it a lot during the pandemic because I was on this Netflix documentary series called Pandemic: How to Prevent an Outbreak.
And I'm sorry I didn't ask you about this. Thank you for bringing it up.
Well, they filmed it in 2019 and it came out on January 20th, right when the pandemic was starting. And so as a consequence, I started getting called, and I was acting as occasional pundit for national news outlets when everyone was trying to make sense of the pandemic. And first off, that was a little alarming. I was like, if I'm the one they're calling, then we have a national communication issue around science. And it was chaotic, right? Some of that's inevitable because not all the facts were known, but like, we should have said that. We should have said, look, we're not sure if the masks help. We know that they help for like flu and stuff. That's why we give them out in hospitals and why doctors. So we recommend people wear them while we figure it out. Instead of, there was this whiplash of, no, they absolutely work, take them, or no, they don't. And I was like, well, epidemiologists are afraid of telling too much to the population because they're like, oh, it'll confuse them. I'm like, no. People are smarter than you give them credit for. What it's going to do is piss them off and make them not trust you if they feel like you're sugarcoating things. And so those of you who remember me on media, I was very careful to stick to my guns on what I think the science actually said. So like when I went out and was talking about the mRNA vaccines, I didn't just go, yeah, they're safe and effective, take them. I said, look, I looked at the data, like these look like they are reasonably safe and effective now. There are some side effects that are, I chose to take them because the side effects of getting the virus, and when I look at both, was much worse than the side effects for my demographic for taking the vaccine. So for me, and I'm an asthmatic, I'm high risk, it made a no-brainer. However, that calculation may not be true for boys age 12 to 17, where we're noticing there's a significant spike in myocarditis and pericarditis. It was about 1 in 5 to 1 in 20,000, 1 in 5,000 to 1 in 20,000 boys in that age range. Like, that's not that rare. In fact, that's about their rate of myocarditis from the coronavirus itself. And so I said that, and I basically was like, look, if I had a boy that range, I might give them something else. Of the other groups didn't have this issue, and I felt like the math on the total, that's the route I chose for me and my family to get vaccinated. So that's a lot more complicated than saying safe and effective, but I felt that it was necessary. And I got shit for it from my epidemiologist friends. Like, Jake, you shouldn't say that stuff. It's going to stoke anti-vaxxers. I was like, no, dude. If you hide that, then people don't trust you. They don't know who to trust. And like my cousins were calling me during that period. And I had people from all over the world who were calling me, and I tried for that first 2 years. I tried answering everyone. It was overwhelming, but people were contacting me, and I tried to just tell them what I know, what I didn't, and here's where the balance was. My cousin's, he's a psychiatrist, and his wife is a surgeon, and they were still calling me, being like, Jake, we can't tell what's going on during media. Do you think these vaccines are safe? And I was like, if they're worried about this, then that means there's a vast silent majority that feels like they haven't been given enough reliable information to make a sound decision. And I think that also that means you need to be more forthright and clear on communication. I think you also need to acknowledge that vaccines are not perfect. So, like, for instance, that coronavirus, the myocarditis thing, we're applying our universal vaccine technology to coronavirus as well as flu. I'm not going to release it unless or advance that program unless I'm convinced that we can address the underlying myocarditis issue, because it's actually, 1 in 5 to 20,000 is unacceptable. Like, I think that's even like 1 in 100,000. You should try to understand what's causing it. And in the case of the coronavirus program, to acknowledge people's concerns, to not tell them, no, you're crazy, take it. People felt that it was rushed. Now, went through all the standard processes, and I took this vaccine. I'm glad I had it. But of course, we were moving faster. It was a pandemic, and they didn't do something that normally happens. Normally, when you make a tetanus vaccine, for instance, you remove the toxic properties of the proteins in the vaccine. And we didn't have time to do that in the coronavirus pandemic. And so the spikes from the coronavirus, they bind extremely tightly to heart tissue because it's the highest expressing tissue of ACE2, which it binds to. And there was no mutation added to remove that effect. And then you see myocarditis. Now, that it's not fully proven that that's the relationship, but it seems a very likely hypothesis. And so my feeling is, I'm just not going to do that with my program. So, we have a program, but I'm like, unless we can convince ourselves that that's the issue, and we have some mutations that have fixed it, but I'm still not convinced that that solves the underlying problem, I'm not going to release that program. And there's a similar concern I have about pediatric RSV, where there's an increased risk of, because we're working on RSV, but then what happened is Moderna got hit with a problem, a ghost of the past from like 70 years ago, where 2 vaccines in children actually made RSV. In adults, it was fine. In children, it made them more likely to get RSV, severe disease. And so I was like, well, until I understand what that biology is, we should just never recommend this drug for pediatrics, because I'm not convinced we understand the mechanism. And then the last thing, just to acknowledge what people are saying, they made a guidance recently about there's a type of mercury that's used as a preservative in old vaccines, and it was put in there. I feel like it was in the 60s or 50s, because there were sometimes batches of vials that went out. Some of them were contaminated with a bacteria, and some people got sick. And so they put a preservative, but they chose this preservative that's based on, it's like thermocerol or something. It's based, I forget, I can never pronounce it, but it's basically I think it's ethyl mercury.
Okay.
And then, of course, in the subsequent decades, we became much more familiar with the problems of mercury posed. Now, in the 90s, they chose to remove this from almost all pediatric vaccines. Like, it's almost no kids are getting this thermocerol. It only shows up in some vials. Even most adults are not getting exposed to it, but there are some products, these multi-use vials. In the United States, it's rare, but there are some cases. It's mostly overseas in the developing world, where they keep it in there because they have a big vial and they dip multiple syringes into it to treat lots of kids or something. And so they use it. Yeah. The administration wants to go in and get rid of it. And I saw some, I was talking, actually, there were people, even there was one person on my own team, and then people outside who were like, I don't know, like, there's no evidence it causes any problem. I'm like, get the fuck out of here. Like, dude, it's mercury. We spent so much time. I was like, and then like there are other preservatives. We have other chemicals. This is just
Yeah,
lazy because people don't want to go rerun the replacement study. I'm like,
yeah, yeah,
get the mercury out of the vaccines. This is a stupid argument for you guys to sit there and be like,
yeah,
there's no evidence, there's a problem. I was like, it's mercury, dude. They claim it's a different like ethyl versus methyl mercury. I'm like, get the fuck out of here, get it out of
yeah.
the vaccines. And so I wrote, I was like, I fully support removal. Honestly, I'm not even that big of a fan of alum, and alum has been safe and everything. It's still an aluminum salt, and I'm like, look, if you have other
Mhm.
options, I'd prefer to use. So I don't have any of that stuff in my vaccines. I tend
Yeah,
to. I'm on the edge of hippie on these things. I'm also.
yeah,
I think what makes it work for a community who doesn't know is you just have to be extra transparent. That's why I did the Netflix documentary. It's why I publish.
sure,
It's and then
sure.
whatever your results are, you have to just tell people straight up, like, here's the range, here's where it's useful. It might be a little more complicated to be like, it's not like when you ask me, it's 100%. I'm like, no, you get maybe up to 95. It's hard to hit that last 5%. Of course, it's an easier soundbite to be like 100% global protection, but that's not true. And I
Yeah,
think if you can spend a little more nuance and just give a little more credit to the intellect of the American public and the global public, then I think you can help reestablish a fractured relationship of trust with the public. And I think that that's really necessary for the mission of this organization, is to be able to move forward. If my intention is to arm future decades with eradication tools to end the forever war against some of our most ancient enemies and make every generation of children healthier, then we have to have a basis of honest communication about here's the range of this tool, here's where it works,
yeah,
and like nobody else does. Like, and you're in the military, someone gives you a gun, they're like, here's where it's a great gun, but sometimes it
yeah,
misfires. You have to tell the
yeah.
limits of the tool, and I
Of
think if you
course.
do that, people are going to be more likely to wield it effectively.
Of course, it's fascinating you're saying this, and we definitely see eye to eye. This whole thing happened while I was running for governor, and it was a hot issue at the
Yeah,
time. And I remember my my only line was educate, don't mandate.
yeah,
Because just like you, I trust the people. I will educate them with the latest data that I have,
yeah,
and then they can decide what they're going to do rather than mandating and then having all of the pushback and all of the issues that you have, whether they're social or whether they're scientific, whatever it was. But thank you so much. Your approach is excellent, and thanks for your honesty on this. We have a lot of work to do, and I hope to get the trust back. And in the meantime, it's up to someone like you and with your Netflix, and someone like me who's paying attention to these things, to slowly bridge the gap and try to make sure we admit what we did wrong, and we now rebuy the people's trust, because at the end of the day, they need to trust us to take your
yeah,
vaccine, which is going to make a huge difference, I believe.
yeah,
So this was incredibly great. I'm so happy that we got a chance to catch up like this in this format. I definitely want to stay in touch. We need to grab a beer sometime and
yeah.
and discuss other things. And by the way, if obviously you have great board members, you don't need me, but if you now had that conversation before, which is like, if on the side you just want to have a conversation about a particular something that you need a non biased person who will have your best interest in mind. You know my number. You can call me whenever you want. You
Awesome,
can discuss whenever you want, and as can Stephanie. So please. So
awesome.
thank you so much, and I hope I didn't take too much of your time. I hear you're out of town, so thank you so much.
Yeah, I'm up in sunny Alaska. Thank you so much. It
Okay,
was this
thanks,
is really
Jacob.
great, and
We'll
thank
talk
you for the
again
offer.
soon. Bye.
Take care. Bye.